Okay, M1 people, I'm going to pick up where I stopped on the lecture a couple of weeks ago and just finish up with the innate responses and then move on to the adaptive immune response against different types of pathogens. So one element of the innate response that I didn't talk about, but which is very important, are innate lymphoid cells, which are present in the mucosa, and they don't detect pathogens directly. They seem to be dependent for their activation on the cytokines and inflammatory mediators that are going to be released by different cells of the epithelium, which are the ones that are actually detecting the pathogens. And then once they get activated, then they'll secrete effector cytokines locally, which allow a rapid response at the site of infection, which is adapted to the threat. So, for example, intracellular pathogens will activate mononuclear phagocytes and dendritic cells by toll-like receptors, Regai-like receptors, um, and maybe the cGAS sting pathway by the release of intracellular DNA from pathogens. So this will lead to IL-12 secretion from the mononuclear phagocytes and dendritic cells. This will activate NK cells and ILC1 if they exist. Also, not to be forgotten is the role of type 1 interferon here, released by plasmacytoid dendritic cells, which is a strong activator for NK cells. So these NK cells, possibly ILC1, will release interferon gamma, and that's important for activating macrophages, monocytes, which will then be able to destroy the intracellular parasites. In this figure, extracellular pathogens and fungi have been considered separately, but it's basically the same thing, because what's going to happen is that the mononuclear phagocytes and dendritic cells are going to be activated by a combination of TLR and CLR, and this will lead to activation, secretion of pro-inflammatory cytokines, including IL-23. And the IL-23 is going to be very important for activating ILC3. And these ILC3 will secrete IL-17 and IL-22, which will have two effects. IL-22 helping the epithelial barrier to reconstitute itself, and also will activate the secretion of antimicrobial peptides by some epithelial cell subsets. And IL-17 will be very important for recruiting and activating neutrophils, which can then phagocytose extracellular bacteria and also Canidia spores from fungal pathogens. So the last type of innate lymphoid cell are, of course, ILC2s. What is kind of interesting or surprising here is that the danger signal seems to come from the epithelial cells, not necessarily mononuclear phagocytes, which will be secretion of IL-25 and IL-33, apparently because due to the damage of the epithelium just by the parasite colonization here. And these cytokines will activate ILC2, which then go and secrete IL-13 and IL-5. And IL-5 is very, very important for recruiting and activating eosinophils. So the right kind of effector cell is able to migrate to the site of the infection and start, fighting against the different pathogens. Now, this basic concept of different groups of pathogen recognition receptors being activated in mononuclear phagocytes and allowing the effector arm to be adapted to the threat is the same thing you see in the polarization of T cell responses by different subsets of dendritic cells. And dendritic cells are, of course, the cell type which connect the innate and adaptive branches of the immune response. So just as a reminder for you and for me, I guess, the main dendritic cell subsets are CDC1, conventional dendritic cells, first type, conventional dendritic cells, second type, and plasmacytoid dendritic cells. So CDC1 are specialized in cross-presentation of antigens to CD8 positive T cells. And they've got TLR3, TLR9 as pathogen recognition receptors, so they can, recognize pathogen-derived RNA, double-stranded RNA, and DNA. CLEC9A is a kind of danger recognition receptor, DAMP receptor, I guess, which is specific for filamentous actin, which normally is just on the inside of cells, but the CLEC9A can allow CDC1 to bind to and be activated by damaged or dying cells. CDC2, the conventional kind of myeloid dendritic cells, which are CD1C positive rather. So they've got a really wide collection of toll-like receptors and CLRs. So these are the ones that are going to be activated mostly by extracellular pathogens. And last but not least, plasmacytoid dendritic cells, which express TLR7 and TLR9. Their main role is to secrete type 1 interferons, the different interferon alpha genes and the other type 1 interferons. Okay, so what's going to happen is, depending on the type of pathogen, then different groups of pattern recognition receptors will be activated, and this will lead to different types of cytokine secretion profiles by the activated dendritic cells. So, CDC1 and to an extent CDC2 will be able to activate Th1 responses by the dendritic cell secreting IL-12. When CDC2 come into contact with extracellular bacteria and fungi, then they'll be releasing IL-23 as well as pro-inflammatory cytokines like IL-1 beta and IL-6, and this will lead to the Th17 polarization of the T cells, which recognize the antigens from these pathogens, which are going to be presented on the surface, or rather, let's say, epitopes from the protein antigens present in these pathogens that are presented in the context of HLA class 2 on the surface of these dendritic cells. Now, one of the things that is still apparently a bit mysterious, this review was from 2021, it seems there's no real Th2 polarizing signal that is comes from CDC2. So I think the current idea is that if dendritic cells come into contact with some kind of helminth antigens, then it's mostly the cytokine environment from the epithelial cells, which is going to prevent these DC from secreting IL-12 and IL-23. And then the kind of default situation would seem to be that if activated dendritic cells present antigen to naive T cells without IL-12 and without IL-23, then these T cells will, and also without the presence of TGF-beta, then these T cells will differentiate into a Th2 along a Th2 pathway. Now, pretty much all pathogens can license CDC2 to activate follicular homing T cell, CD4 T cells, which are going to be important for the humoral response. Now, these T cell subsets, Th1, Th2, Th17, they're going to be secreting the same types of effector cytokines as NK cells, ILC2, and ILC3. So for Th1 T cells, they'll be secreting interferon gamma TNF alpha, which will be important in activating polymorphonuclear neutrophils and macrophages. Th1 responses also are associated with maybe IgG2A specifically and CTL responses. Helminths will be associated with Th2 type responses, which result in class switching to IgE for in B cells, and the recruitment and the activation of eosinophils. Whereas extracellular bacteria and fungi, the Th17 response, the main effectors are going to be, once again, neutrophils and IgG. Now, of course, T cell activation of naive T cells occurs in the lymphoid organs. So this is one of the big differences between dendritic cells and monocyte macrophages that are also in tissues that are getting infected, that dendritic cell activation and maturation results in dendritic cell migration from the epithelial site to the draining lymph nodes. So that's why these are the most important cells for antigen presentation and really starting up the adaptive immune response. Okay, so how about the effectors of the adaptive immune response? And now I'm just going to talk about three things: CD4 T cells, CD8 T cells, and antibodies. And the first of them are CD4 T cells. So of course, these are the cells which recognize peptide antigens presented in the context of HLA class 2. And their role is to orchestrate and orient the adaptive response. So in the lymph node, these CD4 T cells will proliferate and differentiate, and there they will provide help for CTL and B cell expansion. However, after activation, these effector CD4 T cells will also go and migrate to the site of infection, and there they'll secrete effector cytokines if they recognize peptide antigens which are expressed on the surface of the mononuclear phagocytes, which have stayed there. They stay at the site of infection. So Th1 CD4 T cells will secrete, amongst other things, interferon gamma, and this will have an important role in activating mononuclear phagocytes, inducing the expression of HLA class 2 on those cells, and this will also, the activation allows them to kill intracellular pathogens. Th17 CD4 T cells, well, they'll be secreting IL-17, which will attract a lot of neutrophils, which can come in and phagocytose and destroy extracellular pathogens. And Th2 T cells will be secreting IL-4 and IL-15, which recruit and activate eosinophils. Now, the importance of the CD4 T cell compartment in mounting an efficient and effective immune response against pathogens was really shown by, well, many things, but in particular, the effects of the symptoms of clinical AIDS, which occurs when CD4 T cell counts are suppressed below 200 cells per millimeter cubed of whole blood. And of course, this CD4 T cell lymphopenia is a direct result of HIV destroying and killing infected CD4 T cells. So as the disease progresses, patients have fewer and fewer CD4 T cells, and then finally they become susceptible to many different types of opportunistic infections. So pneumonia, so that's extracellular bacterial pathogen, tuberculosis, candidiasis, okay, so tuberculosis, intracellular bacterial pathogen, candidiasis, fungal pathogen, toxoplasmosis, some kind of protozoan parasite, and some DNA viruses as well can become particularly pathogenic in AIDS patients compared to the general population. So CD4 T cells are really, really essential for mounting effective responses against a whole or wide variety of pathogens. However, if you look a bit closer at the different types of T cell differentiation, and you might ask yourself, what is the result if you don't have an effective Th1 response? So there are some patients or people who have a hereditary defect in IL-12 or interferon gamma pathways, and these young children are highly susceptible to mycobacterial and salmonella infections. So Th1 interferon gamma, extremely important for surviving infections by intracellular bacteria. Now, there are also some inherited defects in the IL-17 receptor alpha subunit. So there are people who've got loss of function mutations in this gene in a homozygous state, and these children are highly susceptible to Candida infections and Staphylococcus aureus dermatitis, so extracellular pathogens. Now, I couldn't actually find any information about inherited defects of IL-5, for example. So there doesn't seem to be any severe consequences if people don't have a functioning Th2 response. Now, this might be a bias in the literature because most of these studies are performed in developed countries, or at least countries with some kind of healthcare system with surveillance of these rare disease phenotypes. And I wonder if the load of parasitic diseases and helminth infections in the populations that are studied for primary immunodeficiencies is really going to be enough in order to be able to pick up some kind of severe form of helminth infections that might occur if you don't have Th2 immunity. So that's kind of an open question. So none have really been described, I think, but that's not to say that these effector functions aren't important. Okay, how about CD8 positive T cells? Well, as you know, CD8 T cells, CTL, recognize peptide antigens on HLA class 1. And unlike HLA class 2, HLA class 1 is expressed on almost all nucleated cells in the body. So that means that CTL, CD8 T cells, are extremely important for responses to intracellular pathogens. So activation of CTL by recognizing their antigen on the surface of an infected cell will lead to secretion of interferon gamma and the direct killing of those infected cells by the release of cytotoxic granules, which include granzymes and perforin. Now, you might expect that this means that CTL, CD8 positive T cells are going to be extremely important in the response to viral infections. However, I was kind of a little bit surprised when I checked the types of diseases that occur in people with inherited genetic deficiencies, either in CD8, so there's like one family with this deficiency, or a deficiency in the TAP genes, which are involved in the presentation pathway, involved in the loading of peptide antigens onto HLA class 1. So in fact, the people who have these kind of inherited deficiencies, so they cannot present HLA class 1 associated and cannot present antigen on HLA class 1 to CD8 positive T cells. So these people have problems with lung inflammation, chronic lung inflammation, and opportunistic bacterial infections. they don't have a history of severe viral infections. So that's kind of a bit of a surprise for me. Okay, so for the importance of CD4 and CD8 T cell responses for resistance to a particular type of infection, we don't just have information from the primary immunodeficiencies where rare mutations will have a quite significant effect. But we also have information coming from HLA disease associations. So both HLA class 1 and HLA class 2 are polymorphic loci, and the particular HLA class 1 and HLA class 2 molecules that will be presenting peptide antigens on our cells are going to be different from individual to individual within a population. And depending on which peptide from which pathogen antigen is presented on HLA class 1 and HLA class 2, then the T cell responses against those pathogens will be more or less effective. So that means that if a particular T cell response is a particular type of T cell response is important for controlling the pathogen infection, then it can show up by an association with a particular HLA class 1 or HLA class 2 genotypes. And so there have been a number of studies done to identify whether there are protective or deleterious HLA class 1 or class 2 genotypes associated with a variety of diseases. So perhaps the one that is the best known is HIV. So disease progression, the speed of disease progression and the viral load is strongly impacted by or strongly associated with certain HLA class 1 haplotypes. Well, HLA class 1 alleles, I should say. So HLA-B27 is protective, HLA-B35 is a deleterious allele. There aren't really so many HLA class 2 associations. For hepatitis C virus, there are some HLA class 1 associations, but strong HLA class 2 association. For tuberculosis, no real association with HLA class 1 genotypes, strong association with HLA class 2. So this is telling us, that CD4 Th1 T cells are going to be more important for controlling tuberculosis than CTL. Now for malaria, rather surprisingly I guess, there's a pretty good association with some protective HLA class 1 genotypes, not so much for HLA class 2. For leishmaniasis, it's the opposite. So leishmaniasis is kind of being controlled by the same kind of T cell responses as tuberculosis, for example. So that's what these kind of associations are telling us, okay? What type of T cell response is really, really important for each particular pathogen. Now, one of the interesting things about this is that, of course, with COVID-19, there have been some studies on the impact of HLA class polymorphisms on the severity of COVID-19. So for HLA class 2, the data is pretty weak, and for HLA class 1, generally not clear, but there are some interesting things that have been published recently. So just to give you a kind of comparison between HIV, where there is a very strong HLA class 1 association, and COVID-19, SARS-CoV-2 infection, where there's not much of an association. I'm going to show you this data from a couple of studies in this next slide. So the first one was a genome-wide association study, a GWAS, which was conducted in a cohort recruited by a large consortium of different groups composed of 974 HIV controllers. So the HIV controllers, these are people who, after the primary infection, without any antiretroviral therapy, had a set point viral load of less than 2,000 copies per ml. And the median will probably be about 10,000 copies. So these are people who've got like 5 times less viral load than normal. And these controllers were compared to 2,648 HIV progressors who had a set point viral load greater than 10 to the 4 copies per ml. So in a GWAS study, then a huge number of single nucleotide polymorphisms are genotyped on the genomic DNA of all the people in the cohort. And this was done on Affymetrix chips. And it allows you to screen without any bias for frequent genetic polymorphisms that are associated with your disease phenotype. So this was a big study, and the result was this one. Okay, so each of these numbers down here, this represents like a chromosome from 1 to 22, and this is the the significance of the association with the SNPs, with the single nucleotide polymorphisms at each position in the genome. And what you can see here is you've got like one huge peak of SNPs coming up in chromosome 6. And when you look closer at where this is, these are all around the HLA class 1 locus. So there's a huge impact of HLA class 1 polymorphism on this control of virus replication after the primary infection. Now, that's telling you that the CTL response is the main driver of this, of controlling the virus replication after the primary infection. Now, when this paper came out, I remember thinking, the authors, they must have been so disappointed when they got this result right, because the HLA class 1 associations with disease progression, they'd already been known for 10 years or more in 2010. And I guess they were looking for some kind of new genetic locus, which would have been a new discovery, but they didn't find anything. Now, how about SARS-CoV-2 infection and COVID-19? So, well, of course, this is a GWAS study is a widely used tool. So, of course, there were cohorts set up to try and test and find out what are the genetic loci important for disease outcome. So there were a few of these studies done. This was one of the biggest ones with 2,244 cases who were patients with severe COVID-19 and 1,676 controls who hadn't been infected. So this is, the global general population in the UK versus the patients who had severe COVID-19. And these were the results, with a few peaks coming out. But if you looked on chromosome 6, there was no HLA signal. So that's telling you that the T cell response is not really the main driver in determining whether someone who is infected with SARS-CoV-2 is going to develop a really severe life-threatening infection. It's not CD8 T cells, it's not CTL that are, involved in this either severe disease phenotype or better control of virus replication, which prevents people from getting severely ill. But some of the SNPs that came out were in genes. OAS1, it's an antiviral effector system. Interferon alpha receptor 2, TYK2s, the signaling tyrosine kinase for the interferon alpha receptor. So these are confirming that it's a type 1 interferon response that's really critical here, that determines whether you're going to have a severe infection or not. Okay, so that's where we stand with SARS-CoV-2. There's been some interesting work published just last year, actually. So CTL response in COVID-19 SARS-CoV-2 infection doesn't seem to be related to severity of a symptomatic infection. But here there's a paper published by a group who were interested in testing whether some particular HLA genotypes might be associated with asymptomatic infection. Now, that's something that's not so easy to study, because the people who've got an asymptomatic infection, they're never going to go to the doctor, right? So how do you even recruit a cohort of this kind of patients? Well, there are different ways of doing this, and the one that was done here, which is something I think was really clever. In fact, the authors recruited a cohort of bone marrow donors. That's important because bone marrow donors already have their HLA genotype, which is done. So they contacted all these bone marrow donors in the database and said, we want you to participate in a citizen science study. And this was based on smartphones. So the people involved in the study had to download the app. And the app was there to track their COVID-19 symptoms and outcomes. And mostly self-reported symptoms. Have you got a fever? Have you got a headache? And whether people had a positive test. So the recruitment was carried out from July 2020 to April 2021, before vaccination became widespread. And I think this was mostly done in the US, in California. And by that time, they had got results from 1,292 people who had a symptomatic COVID-19 infection. And they had also managed to pick up 136 people who had an asymptomatic infection. So they had a positive test for, an antigen, rapid antigen test, but they didn't have any symptoms, okay? No fever, no tiredness, no headache. And they looked at the HLA genotypes in these two groups of people, and they found that the patients with an asymptomatic infection had more frequently this HLA genotype, HLA-B15. So there were 20% of the people with an asymptomatic infection had HLA-B15, but only 9.4% of people with a symptomatic infection were HLA-B15 positive. So this was an odds ratio of 2.4, highly significant. Now, why was this happening? What was the physiological basis of this? So HLA-B15, the authors reasoned that this molecule, HLA class 1 molecule, will be presenting some kind of particular peptide from SARS-CoV-2. So, excuse me, I'm just letting my dog out of the house, so I'm going to be coming back to the screen in a minute. Okay, that's it. He's got out the front door. I'll probably have to go and let him back in a little bit later. Okay, so they reasoned that HLA-B15 is having an effect because it's presenting some kind of epitope from the SARS-CoV-2 coronavirus. And so they tested different peptides which displayed the binding motif to the HLA-B15 molecule. And what they wanted to know was whether the patients, or these people rather, they weren't patients, they weren't ill, whether the people with an asymptomatic infection had some kind of CTL response against the epitope before they had actually encountered the virus. So they got PBMC samples from this cohort of patients because they're all bone marrow donors. They maybe had some frozen PBMC. So they found PBMC, peripheral blood mononuclear cells, from before the pandemic, and they were able to test those PBMC for a response, or for the presence of CD8 positive T cells that recognize these different HLA-B15 restricted epitopes. And sure enough, they had, for this particular epitope, which they call NQK, they found 1, 2, 3, 4, 5 out of the 9 people they were able to test had a detectable CTL response against this epitope. And that was before SARS-CoV-2 had even entered the human population. So how come these people had a CTL response against SARS-CoV-2 when the virus didn't even exist? Well, it seems like these CTL were cross-reactive. And they were cross-reactive for the same epitope from seasonal beta coronaviruses, which are infecting us every year. So basically the idea is that this HLA class 1 allele was associated with asymptomatic infections because it was able to present an epitope that was the same between seasonal coronaviruses and SARS-CoV-2. And this allowed people to have an asymptomatic infection. So people would get infected, but in fact wouldn't get ill. So it would prevent illness. So if you had a pre-existing CTL response against SARS-CoV-2, this seemed to be related to milder disease course. So that's a fantastic story. So that was why it was published in Nature. But it seems that this HLA-B15 association with asymptomatic infection, which is at the base, at the start of this story, hasn't really been confirmed in an independent study. So you have to look out in the future for this one. Okay, so that's SARS-CoV-2. Now I want to go back a little bit to HIV infection. So this is one case where there was a really, really strong association between HLA class 1 genotypes and the viral load set point after primary infection. So that's suggesting that CTL are extremely important for controlling virus replication. Now, in humans, it's not really feasible to get data on primary infection because people don't know when they're getting infected. They know they got infected maybe 1 month later when they get a seropositive test. So that's why animal models were very important here. So in the 1990s, there were a lot of groups that developed models of SIV infection in a variety of different animals, mostly in macaques. So here we had, I'm showing results from a paper published in 1998, where they had like 3 animals infected with SIV, either sooty manga base or rhesus macaques. Now, what you want to look at here, the empty squares here, the white squares, that's viral load, okay, by PCR. And the different dark lines here are CTL responses. So you can see that pretty much in every case, well, 5 out of 6 in any case, you've got the peak of viral load and it starts to go down just as you get the CTL response that's kicking in 2 to 4 weeks post-infection. So this is telling you, when do you have CTL detectable in the peripheral blood? You don't get a CTL response 1 week after infection, 2 to 4 weeks maybe. Now, so that's pretty good. Looks like as the CTL response goes up, viral load goes down. So is it possible to prove that it's the CTL which are controlling the virus in this case? So this was another experiment which was done because it's possible to deplete CD8 T cells with antibodies. And so this was done in a group of macaques. So they had 6 macaques as controls and 6 which were depleted with anti-CD8 antibodies. Now, out of those 6, only 3 of them had a durable depletion of their CD8 T cells, and 3 of them had a kind of temporary transient depletion of CD8 T cells. So this is viral load again, going down between 2 and 3 weeks after infection in these 2 groups, stays up in the group of macaques where the CD8 depletion was really durable. And here's another example of the clear squares here. This is our detectable CTL against SIV, and the triangles, this is the viral load. So as the CTL come up, the viral load goes down. And in the macaques where you don't have any CTL response, well, the viral load stays up. Okay, so this is telling you that it's really the CD8 T cells which are responsible for the control of virus replication in this case. Now, in terms of clinical progression, what happened? So the 3 animals with a severe CD8 T cell depletion, well, they were all dead in 6 months. So in the absence of any CTL response, viral load, virus replication is not controlled in this post-acute phase. and that leads to accelerated disease. Okay, so that's CD4, CD8 T cells, and now we're looking at the last thing I want to talk about, which is the humoral response. So antibodies. Now, most antibody responses to pathogens are T cell dependent, so they are developed in the B cell follicles of secondary lymphoid organs. They're dependent on help from CD4 positive T cells. So the naive B cell, which expresses an antibody on its surface specific for a particular antigen, will internalize that antigen, process the peptide epitopes, and express them in the context of HLA class 2, which can then be recognized by the helper T cell. Oh, excuse me, I'm going to go let my dog back in. Be back in a minute. And so the development of the T cell dependent response will lead to isotype switched high affinity antibodies, and long-lived memory B cells and long-lived plasma cells. So you get IgG, IgA, and IgE. So the types of antigens that are producing this kind of response are basically bacterial exotoxins, viral envelope glycoproteins, individual proteins, either associated with bacterial cells or pathogen cells or particles, or secreted. For T-independent antigens, then you can develop an IgM response composed of low affinity antibodies, and these IgM secreting plasma cells develop in the extrafollicular compartment. However, they can lead to the production of IgM memory B cells. So this can be a kind of also a long-lived memory B cell response. Now, the types of antigens which lead to this T-independent response, I'm going to have to go let him out again, excuse me. Okay, so the types of antigens lead to T-independent responses seem to be when you have multimeric antigens. So you've got a dense array of repeated epitopes on the same kind of particle. So typically these are bacterial capsule antigens. Also, some of the very dense glycoproteins on the surface of particular parasites like Plasmodium falciparum merozoite antigens, they give a strong IgM memory response. And in the lab, we've got some indication that some non-envelope virus capsids can also give you a pretty significant IgM memory response. So yeah. Now, when does the humoral response kick in against a pathogen? Now, SARS-CoV-2, the COVID-19 pandemic, was a kind of opportunity for every immunologist to re-demonstrate the classical aspects of immunology in a new infection. So one of the things that was done was to track the development of the humoral response after an acute infection. So this was one study published in 2020, and some typical results here, looking at the titers, the ELISA titers of IgM and IgG against the spike glycoprotein. And over time here, this is time after symptom onset. So you can see that, IgM in the dark symbols is coming up sometimes a little bit quicker than IgG, but then after the peak, IgM is going to go down and IgG is going to persist for longer. So here, generally speaking, the patients had seroconversion 2 or 3 weeks after symptom onset. So you have to think this is probably at least one week longer after infection, because you'll have an incubation period of maybe 6 or 7 days for SARS-CoV-2 infection. So the seroconversion is coming, 3 to 4 weeks after infection, 2 to 3 weeks after the onset of symptoms, and IgM coming up first. So this is really, classical humoral response to an infection. And the other thing that they showed in this paper, and several other groups showed this, is that the more severe the infection you had, the higher the titer of antibodies. So, a mild asymptomatic infection, you're going to have a low antibody titer, might not even seroconvert. Patients who were hospitalized, really, really high titer of antibodies. So that's during the acute phase. How about B cell memory? So this was one of the studies from Shane Crotty's lab. They followed patients up to 6 months post their infection. So this is ELISA titers of spike-specific antibodies. So after infection, it's going up, and then slowly it's waning. Okay, but IgG memory B cells, they persist over a much longer time period, whereas the IgM memory B cell compartment, they're shorter lived. Okay, so antibodies, seroconversion, 2 to 3 weeks post-symptom onset. Okay, what are the effector functions of antibodies in an infectious context? Well, there are different kinds of effectors associated with antibodies, which are more or less efficient at controlling different types of pathogens. So here on my little kind of diagram here, the key to understand this is the pink. These guys are gram-negative bacteria. This purple thing here, that's gram-positive bacteria. This is a protozoan parasite, and this is filamentous fungi, and this is a yeast infection. And down here is a virus. So getting back to the effector functions of antibodies, opsonization, very important, okay? Antibodies binding to the surface of a pathogen will allow it to be phagocytosed by cells which carry Fc receptors, mononuclear phagocytes, and also neutrophils. Complement deposition, also very important for opsonization. So if you've got C3b on the surface of a bacterium or a parasite, then once again, cells with phagocytes bearing complement receptors can internalize this particle and destroy it. Okay, another effector mechanism related to antibodies is, and related to complement, is the formation of the membrane attack complex on the surface where antibodies have bound. So antibodies bind, IgG, IgM bind. This will allow the activation of the classical complement pathway, formation of the membrane attack complex. And this is effective against gram-negative bacteria and protozoan parasites. Opsonization effective against gram-negatives, gram-positives, protozoa, and fungi. The third mechanism of effector mechanism is neutralization. So that means that antibodies are binding to the adhesins or the attachment proteins and glycoproteins that pathogens use to attach to cells and evade tissues. So if you block these adhesins or attachment glycoproteins, you prevent the infection from occurring. So this mechanism is effective against gram-negative, gram-positive bacteria, viruses, and protozoan parasites, not so much against fungi. And the last mechanism, also very important, is inactivation of toxins. So very important to prevent disease for some bacterial infections. Now, the type of effector mechanism which is important depends on the pathogen, okay? And the reason why a vaccination works depends on which is the effector mechanism which is being mobilized. So, for example, pneumococcal vaccines, after they're composed of multiple different capsule antigens, you get antibodies against these capsule antigens, and binding of the antibodies to the surface of the bacteria allows them to be phagocytosed and killed. So that's the most important effector mechanism for resistance to Streptococcus pneumoniae. Whereas meningococcal vaccines, Neisseria meningitidis, then what is important here is direct killing by complement. So meningococcal, so that's Neisseria meningitidis, gram-negative. So it's more sensitive to complement-mediated lysis than gram-positive Streptococcus pneumoniae. Neutralization, blocking adhesives, like basically all antiviral vaccines work by this mechanism. It's also the case, that's the mechanism that functions in the recently licensed anti-malarial vaccine based on the circumsporozoite protein. So it's binding to the CSP, which is going to prevent the infection of hepatocytes by the sporozoites that are injected. And also it's a mechanism for some antibacterial vaccines. And finally, diphtheria and tetanus vaccines work by inactivating the bacterial exotoxins. Okay, so that's finally the end of this lecture. So just to wind up here, you have different phases of the anti-infectious immune response: the immediate phase, the early phase, and the late phase. So immediate response, this is all to do with detection of pathogens by different pathogen recognition receptors, different PRRs, which are going to be different depending on which type of pathogen. The early responses, we're talking about hours to the first few days post-infection. This is where the innate response is really important. Determines whether you live or die and how severe the infection is going to be. The type of innate effectors are going to depend on what type of pathogen was detected in this initial phase. And the late phase, well, 4 to 14 days, maybe after the first week of infection, then you can get some CTL, some detectable T cell responses, antibody responses. We're talking about 2 to 3 weeks post-symptom onset. So adaptive T cell responses, antibody responses, and this is going to be important for clearing up any pathogen that's still there, eradicating the pathogen at the site of infection, any systemic infection, eradicating the pathogen all around the body, and also essential for acquiring immune memory in order to have a more effective, more rapid response to future infections and reduced pathogenicity, reduced morbidity. So functional immunity to infections depends on your memory T cell and memory B cell responses. Okay, and with that, I'm going to go and finish the recording and let my dog back into the house. Okay, thanks for listening.